Hi, everyone. This is Nick Langhals, Program Director for Neural Engineering at NINDS here at the NIH. I'd like to thank you all for joining us for the first Brain Initiative webinar series on technologies for recording and modulating neural activity. We have invited two speakers for today. Loren from the University of California, San Francisco will be speaking on technology for understanding distributed brain circuits. And Isacoff from University of California, Berkeley will be discussing imaging and manipulating synaptic transmission and plasticity. The format of these presentations will be a few minutes of a kind of general, broad overview of the work that the investigators are doing, followed by a more detailed discussion of the current state of their technology that they're in the process of developing. The total time for presentation, each presentation is allotted to be 20 minutes, followed by a 10-minute discussion section. For the discussion section, there will not be an open question and answer. We have nominated two different discussants, Lynn Tien and Cindy Chestek, who will coordinate and ask questions and lead that discussion over the topics with the investigators, as well as the two investigators will be open to asking questions related to each other's work. We hope that all of this is very useful and a good endeavor, but this is our first one in the pilot, and so there are technical difficulties. Please let us know afterwards so we can do our best to correct them for the next one. So at this point, I'm going to go ahead and turn it over to Loren to do his presentation, and hope you all enjoy this new feature.
So thank you, Nick. It's a pleasure to be here, and I will try out. Okay, I can see myself, which is great, and I will share my screen in a moment. So what I wanted to start with, because this is the first one, was a quick sort of overview of, from my perspective, why I think this really matters and why the work the Brain Initiative is supporting actually is important, hopefully, to everyone at some level. And, you know, for me, it sort of all comes down to the fact that who we are, everything that we do is because of the sort of 3 pounds of gray stuff up in our heads. And it is this massively complex thing made up of something like 100 billion neurons, individual brain cells that connect each of them to maybe 1,000 or 10,000 other cells. And it is fundamental to who we are, and yet, at the same time, we are also profoundly ignorant about how it works. We just don't understand it. And that's a problem. And it's a problem not only just because, for those of us who are scientists, we'd like to understand things, but also it's a problem because when it breaks, we don't know how to fix it. And this is a serious issue for us, both personally in our country, things like schizophrenia, stroke, Alzheimer's, and so on, both at a personal level. These things have huge personal costs, but also at an economic level. Alzheimer's alone could bankrupt this country in 50 years if we're not able to do something about it. And the problem, from my perspective, is that we have all of these things that go on in the brain, and we can't fix it. We can't do much for people with these problems, in large part because we don't understand how it works. And it's really hard to fix things when you don't understand them. And so what I see this as doing is sort of recognizing that we need to understand how our brains work to help people live more productive, effective lives, to fix the brain when it's not working well. And that's really what this is about, is developing the tools to make that possible. And in our particular case, we're working on tools to measure activity in the brain, right? The brain is made of these tiny little neurons, and one way to do this is to put electrodes down near them and to measure their activity and to see what they're doing. But the current technologies let us get relatively small numbers of these neurons and make it hard to do more than a set of neurons, kind of one part of the brain. And so what we're really trying to do is build a new way of doing this, where we have flexible, soft electrodes that play nicely with brain tissue, groups of these electrodes that we can put wherever we want to in the brain, and then a whole system going all the way from these electrodes in the brain to new computer chips and hardware that takes these signals and sends them out, new processors of it, new software that's freely available to everyone to actually let us look at these things, and also hopefully to, you know, actually drive a lot of new science in understanding how the brain works. So that's sort of the brief overview. I will go ahead and share my screen, and hopefully this will work happily. And then I'll now go into sort of more of the details of what we're trying to do. And so one of the things I want to emphasize here is that this is at a very fundamental level a collaborative effort. And so that's the other thing that I was really excited about with the BRAIN initiative, is it really focused on bringing groups with different patterns of expertise together. And so this is what we've been able to do. So we've been working with Vanessa Pelosa, who's a very talented engineer at Loren National Laboratory, and her colleagues, and they have developed some really amazing techniques for column lithography that I'll talk about. We're also working with Reed Harrison, who is a former academic, now in the commercial sector, who builds chips for taking neural data and amplifying it and multiplexing it and doing all the other things we need to do to get it off the head and into our computers. And finally, we're working with another company, Spike Gadgets, which happened to be founded by my first graduate student, Matthias Carlson, and his father, Magnus. And it turns out that Magnus is a really amazing digital engineer. And so what this has let us do is bring all of these groups together to do something that none of us could have done on our own by any stretch of the imagination. So I first want to start then with just what is the problem? What do we want to be able to do that we can't yet do? And roughly it corresponds to this, is that what we have right now, we can either record a reasonable number of neurons in one part of the brain or a few neurons in a bunch of different parts of the brain. And this is a problem, like for my own ongoing work now, we're interested in how different parts of the brain interact during memory storage and retrieval and decision making. And it's really hard for us to get a good enough sense of what's going on to really fundamentally answer those questions. So we have things like these really beautiful multi-tetrode microdrives where we have 4-wire electrodes, and each of these is one little microdrive that can move it into the brain. But they're pretty big, and you can only put so many of these on the head before you run out of space and before it's too heavy. And we have connectors. This is, 13 millimeters, which is about a little over half an inch, and that sounds small, but if you want to have thousands of channels, that's a real problem. And even the microchip, so this is the state-of-the-art microchip in the bottom right here from Implant. These are great, but they're too big and too power hungry for us to do this for thousands of channels. So in particular, we have these limitations to really scaling this up to being able to measure more than a handful of neurons. The head stages, the things that take the data from our electrodes and send them out, are either too big and require too much power to scale up to thousands of channels. The connectors, and this is actually an amazingly sort of boring but incredibly important problem, is just getting the data from the electrodes to the chips. Often you use a regular connector and they're too big. So if you want to do thousands and thousands of channels, you have to have thousands of channels of connectors, and pretty soon it's huge. Furthermore, we have a hardware and software problem. Buying a standard commercial system that supported 1,000 channels would be extremely expensive, and this, in this era of lower NIH budgets, is a real challenge for many of us. And so our goal was to try and solve these problems. So specifically, we want an integrated system that can do the following things. We want to be able to record from thousands of neurons. That is, we want to be able to record from at least, 10 times as many as we can do now. And this is the first step, and we'd like to go to 10 times as many than that further on. We really want to be able to flexibly distribute recording sites based on experimental need. And I just want to talk about this for a moment because it's actually really important. Other efforts which are fantastic that are, for example, developing electrodes, are developing electrodes with very high density recording in one region. And that's a very important thing to do because it's really important to understand how local neuronal networks do stuff. But at the same time, every single thing that our brain does is based on a massively distributed network of neurons. It doesn't happen just locally. And we need tools that allow us to measure those distributed patterns of activity, to understand how activity goes from one place in the brain to another place, and so on. And this is different for different experiments. So we really want to be able to put electrodes where we need them when we need them. We also would like to do this for a long time. Most current recording technologies are pretty limited temporally. Often you can record for a couple weeks or a month. And if you want to understand, for example, developmental progressions from early childhood into adulthood, we really want to be able to record continuously for months or years. And that would open up a whole new set of questions that we've barely started to think about. We also want to record, at least for the moment, in a variety of awake behaving animals, but also I'll say, the nice thing about the technology we've been using is that it's something that's compatible with the human brain. And then finally, we also want to do all of this at high resolution, both in time, that is in milliseconds, which is the time scale on which neuronal signals exist, and in space. And for us, that's the single neuron resolution. For others, there are even higher resolutions that are relevant. Okay, So how are we doing this? So what we've done, as I said, is to bring together a number of people with different expertise to develop surgical methods, flexible probes, miniature head stages, and open source software. And I'm going to briefly go through each of these things to tell you what we've done and what we're doing. All right, and as you can see by all of the little logos, this is really a joint project. This depends on coordination across a number of sites, and we've been really happy with how well that's gone so far. So let's start with the flexible electrodes. Now, this is a key element of it. This is sort of a cartoon of an earlier version of the design. I'll show you a later version in a moment. So basically what we have here is a connector pad. This is for a connector that we can use to get the signal out. And this is sort of over here where my mouse is. This is the business end. This is the part that actually goes into the brain. Right now it's 6 millimeters long, but they can be made longer. So here we have 2 shanks, each of which in this older version had 16 little recording sites, each one 20 microns across. So that's about 1/4 the diameter of a human hair. And we were recording across about 380 microns. So now the really nice thing about what Livermore has perfected is these are very soft and flexible. And so compared particularly to materials like silicon, these, we think, can move with the brain more easily. And particularly in larger animals and in humans, this is a big deal because the brain moves a lot inside the skull. So we really want a technology that can float along with the brain. And we also have done many design iterations. So we've gone through multiple versions. As I said, this is an older one. I'll show you the other one. And this has really also been critical to getting something that works because we have to communicate between the engineers and the scientists. This is a layer-by-layer fabrication process for the aficionados, and it's a thin film polymer. So it's layers of polymer that are a micron or 2 thick with layers of metal. So this uses sort of standard lithography, in this case photolithography techniques, to make this all possible. And the other thing that's important is because this is a fairly potentially standard thing, once we get this working well enough, it's something that we hope to be able to distribute to lots of other labs. And indeed, fortunately, lots of others are interested. So we're trying to figure out how to do that right now. Okay, so now one of the issues here is how do you get these things into the brain? Because they're soft and flexible. And so here the group at Livermore built a really, from my perspective, a beautiful technology for making this possible. They take a silicon stiffener, and that's actually what you're looking at in the background here, and this is a little puddle of polyethylene glycol, which is basically we use here as a biocompatible adhesive. And so they heat it up, and there's a silicon channel down the stiffener, and you can see it wicking down that channel just thanks to surface tension. And then they micro-align the polymer, which is this thing on top of the silicon below it, and then down this little channel wicks the thing. And then when they're done, here's an example of that. They put it into something soft, they can pull the stiffener out, and you can leave the electrode in place. And so as a result of this, using this and adapting this for animals, we've been able to put in large numbers of these probes to have them stay where we leave them and to get really high quality recordings. And that's what I'm going to that's what I'm going to illustrate here. So we put these in the prefrontal cortex of rats. And this is sort of a cartoon, again, of what the tip of one of those electrodes look like. And in the lower right, you can see the actual probe. So again, these little black dots are the recording sites. And we put this in on day 1, according to this particular timeline. And on day 45, so a month and a half later, we were able to isolate a total of 18 separate neurons. And you can see here, each of these shows the waveform of a single neuron recorded on the multiple channels indicated by the color. And these are the sizes, which are pretty respectable for neuronal signals. And these are 18 sets of neurons recorded on day 45. This is that same probe on day 154. So we're about 5 months out now, and it's not quite as good, but it's close. And here it is on day 283. And we stopped the experiment a little bit after this at 10.5 months, not because the probe was failing, because we were worried the animal was failing because it was reaching the end of its normal lifespan. And so we're really excited about this because, if you imagine you're trying to understand how, for example, the brain learns and remembers and changes with aging, these sorts of technologies have the possibility of carrying you through the entire lifetime, basically. All right. So that's the older generation. Here's the latest version. Now, I apologize for the image quality. This is something my graduate student took a couple of weeks ago, so it's really fresh off the press as a way. So this is a new 64-channel probe. So if you look here zoom in on the tip, and obviously this is taken in a different time, but this is what it looks like. You have 4 shanks. Each of these is 68 microns wide. So again, about the size of a human hair, but only 14 microns thick, with 16 electrodes, and there are multiple layers of metal contacts. And those go up to this PC board. So this is like a board you'd find inside a computer, but this has a couple of important characteristics. So this is the zoom of it. First, over here is the bond pad. So I mentioned before that connectors are really a problem. Getting all of these signals out is a problem. So our colleagues at Livermore developed this specific bonding approach where they can now bond in 2 dimensions instead of 1. And so basically we have 64 bond sites in a 3.5 by 3 millimeter grid. And you can see there's lots of space for more bond sites. This means that basically we've gotten rid of that connector problem, and we now have the capacity to really think about thousands or even tens of thousands of channels, and that's really important. We also have the current state-of-the-art 64-channel Implant chip, which you can't see because of the contrast, and then a stacking connector that allows us to put a bunch of these together in sort of a flexible configuration. And that's kind of what this looks like. So this was taken just a few days ago. This bottom board here, you can see the scale here. So this is 1.5 centimeters high and about 2.5 centimeters or about 1 inch wide. This is an FPGA board. This takes all the signals from a stack of these boards, puts them into a single digital stream, puts them out through a 12-wire micro HDMI connector that easily handles 1,000 channels. And we've tested that and it works. And here we have, in this case, 3 of these boards. These are the polymer ribbons coming off those boards, so they stack. We can have a stack of up to about 8 of these. This is just a terminator board. And so each of these is a 64-channel module. And this is the module sort of in the background. If you imagine another module connected to this connector here in the foreground, this would be 6 of those boards times 64 channels, or 384 channels in a 1.5 by 2.5 by 2 centimeter package. This, as I said, the electronics works, it's small, it's light, and it's something, because each of these is separate, we can target each of these electrodes to specific regions of the brain. And that's really what we're excited about, is this idea that we can finally measure activity across these distributed circuits in a way that we never could before. Okay, so that's still, however, not enough. We need to get these signals to a computer, and again, this is where working together on all of this has really been so productive. So we have these polymer electrode tips. Those feed into these head stages. So again, each of these little things here is a 64-channel head stage board. So this is one of the, our older versions of it. That goes via this 12-wire cable to this thing, which is about the size of a paperback book. It's made by Spike Gadgets. And here, I would just want to say, partnering with a company has been great because it means they have a product that other people can use, and they stand behind their product. So this private-public thing has really been critical for us. And then, thanks to experiences I had with previous generations of data acquisition systems that I helped design and set up, we decided to use Ethernet. So this is what you plug your computers into the wall with, packets. And these packets are really fast, and this is an incredibly robust way of getting information around. So basically, you take this main control unit, plug an Ethernet socket into the back, plug that into your computer, and suddenly you have your data. And then, finally, you can then look at your data on a screen. And I'll just spend the last minute or two just talking about that part. So we built, and really this is a lot with Matthias, my former student and now one of the co-founders of Spike Gadgets, a cross-platform software package. Whoops, sorry. This is what it looks like. So each of these traces would be the continuous trace from one tetrode. These little diblets here are spike waveforms. You can look at projections of that activity, so the amplitude on one channel of, in this case, a tetrode, 4-wire electrode versus another. You can see the individual spike waveforms and so on. This works for Trodes, for as many channels as you'd like on any individual one. And this software has a couple of things that are important. The first is it's written by actual electrophysiologists, which is useful because we sort of know what we want. And it's open source, which is another key. I, like many scientists, am a complete control freak. And I found that being able to use my own software and being able to modify it has let us do experiments that we couldn't do any other way. So we really want this to be a resource to the community, if people want to use it, to be able to do their own experiments. The software is modular, meaning that it runs with a bunch of modules that communicate with each other, which means that people can reasonably easily write new modules to do new kinds of analyses. And it's cross-platform, so it runs on Linux, Mac OS, and Windows. It uses something called Qt, again, for the aficionados. So this allows for a couple of things. First of all, it works for 1024-channel data acquisition. We can stream that to disk and visualize it. Obviously, you need a fast computer, but that's not so bad these days. You can look at the data as you get it. You can control experiments. That's another thing that Spike Gadgets has built as part of this. You can also take video data, and so we're excited about the idea of also joining this with, for example, imaging, so that we can get simultaneous electrophysiological and imaging data sets. And also, this is really critical for a lot of what we want to do. This allows for real-time feedback. So we can currently take a signal from the animal's head, get it to software, process it, and then send, say, a turn on a laser or a stimulation pulse back to the animal's head in less than a millisecond. Our median time is 0.8 milliseconds. And that's, again, because we took advantage of this Ethernet architecture, which is really fast. And that means you can start doing experiments where you use the content of brain activity, what the brain is doing, and perturb that content to understand how different patterns of brain activity actually do things. And for reference, this, as I said, it's open source software, so this is the URL where it can be downloaded from. If you just do a search for Trodes, T-R-O-D-E-S on Google, I think it will come up. And there's a Bitbucket repository and lots of compiled binaries for people. And again, it's open source and free, so we're hoping that people are interested in using it. All right. And then finally, I just wanted to end with, where is this going? Well, to the extent that we would like to help people, we would like to build systems, and this is this kind of cool graphic from Livermore, that can interface with the human brain. And what's neat about these polymer electrodes is they've actually already been used to interface with the human brain in the context of an artificial retina project that was done by the Department of Energy. And these are things where we could actually put these potentially someday in a human being to help monitor patterns of activity and perhaps to help correct those patterns of activity when we needed to. All right. And then finally, I really, again, need to acknowledge the team. So there's an entire team at Lawrence Livermore National Lab. This is Vanessa again, and Kedar, Angela, Sarah, Supin, Elaine, Charlie, and Aaron, who all did important things. In my lab, I have 2 amazing graduate students and others who have worked really hard on this. But Jason has really pioneered the development of all of this and getting this working on the animal. Daniel has been incredibly important for that, as well as the software. And then the father-son team of Magnus and Matthias Carlson, who made all of the electronics and software possible with a little bit of help from me and others. And then also Reed at Intam. And again, I would just like to say none of this would have happened without support from the BRAIN initiative. The funding has made it possible for us to do this. But I should also acknowledge that this all started with a smaller grant from the University of California, sort of a high-risk, let's join up the UCs with the National Labs grant, which we're also very grateful for, and I now have some continuing support from HHMI. So that's it. I'm happy to take any questions.
Cool. Thanks a lot, Loren. So to start off the discussion, we're going to go ahead and turn it over to Cindy Chestek to ask some questions.
Hi, Loren. Great presentation. So I'm very intrigued by, it seems that you guys are making a direct connection between your probes and the board that has the chip on it. So does this mean we have entered the era of disposable integrated circuits?
Yeah, we've been debating that a lot. Is it worth me going back to that slide to illustrate it, or what would be helpful for people?
Sure.
Okay.
Yeah, let's see it. Yep.
Okay. Yeah, so let me go back to that. So that's here. So, we've debated this a lot. So the probes themselves are pretty expensive. And the chips are expensive, and so this all gets to be very expensive. And so the question is, do we just throw these out after we use them? So our first thought is we can actually implant these, and we embed this entire ribbon in silicon when we do it, so it's soft and flexible. So we're reasonably sure that we could recover these devices even months after they were used. So that's one answer. A secondary answer is if we're using these devices for data collection for months, then it starts to be less financially prohibitive because the amount of data you get from each one is so huge. If you figure it out, if we're taking, we can actually think about taking continuous data now from 1,000 channels. That amounts to 60 megabytes of data a second, which is 15 terabytes a day, which is starting to make my head and my wallet hurt, but it's still possible. But finally, there's another thing which I'm really excited about, which I didn't mention just due to time, which is the new Intan chip that Reed is building. It'll be a little bit bigger than this current one, but it'll be 128 channels, and it has what's called, sorry for the technical parts, ball grid array packaging, which means it's just a bunch of little gold balls on the bottom, which means it's actually possible to solder and desolder those expensive chips from these boards. So I think what we might actually be is...
Sure, sorry, is that going to be reasonably priced, though, to pull a BGA and put it back on?
Yeah, well, people can do it at home with a hot air thing. Okay. So, it's doable. This is all very experimental, so I don't know what's going to work, but I think it's possible.
Okay. So another question that I had. So So now that you guys have done a bunch of these experiments, I noticed that you sort of, noted when you thought it was the same neuron on different shanks. If ultimately the number of wires that you can get out of the brain is your most scarce resource, what do you think is the maximum effective density if you kind of want to get, on the order of one independent neuron per channel?
Yeah, it's a really great question. So there's, so what we see here, and I'll just go back to that quickly, is, these are channels that have a 20 micron edge to edge or a 40 micron edge to edge spacing, and that seems to be pretty good in the rat, which is where we're doing this, for getting a large number of cells. Now, there could be smaller cells that we're missing, but this seems like a pretty good density as far as we can tell. And, ideally, we'd like to carry this further up the shank, but it's not clear to me that going much higher than this, at least in the cortical area that we're recording from, matters. That said, when we've tried these same ones in the hippocampus, we found that they're actually not dense enough there, and that's an area where the cells are very densely packed. So we think that, in fact, you need smaller contacts and denser packing for some brain areas where the neurons are more densely packed, or similarly for mice where the neurons are smaller, you probably would optimally want those. But we're sort of in the ballpark here. 50% either way would probably be good for a whole bunch of systems. And then there's a broader question about, how densely can these be put in the brain before you start doing damage? And I don't know if you wanted me to comment on that.
Do you have any initial data on that?
Yeah, so these we don't get a lot of damage. It's hard to see the tracks of these things even after months, so we're sort of optimistic. And, again, because we're seeing neurons even 10 months out or, 9 months out, that suggests to us that we're certainly not trashing the tissue. In general, what we're comfortable with now is about a 250 micron spacing between these shanks. And we're also, as you may have seen, when we're working with smaller shanks. So in principle, we can imagine, putting these in at 250 micron spacing all over the place, and that gets to thousands of channels pretty quickly. Sure.
Okay, so maybe, so then let me ask a question about for everybody who's excited about getting these. So is this, eventually the vision to have this whole thing distributed as a unit that ships to labs, and is Spike Gadgets going to provide that full holistic electrode chip spiking unit?
Well, so right now the bonding, so what we have right now is the probes are manufactured at Livermore, and they have a facility that's great for that, their own sort of biocompatible clean room. Spike Gadgets makes the boards and ships them to Livermore for that bonding process. And so we are really trying hard to figure out how to do this and how to make it economically viable for everyone. Part of the problem is that, even just making these at Livermore, it's an expensive process because that's an experimental facility where they're really trying to push the boundary. And so we're asking, can we outsource this? And then could we outsource the bonding process as well to someone? And we don't yet know how to make that work. So we are very open to ideas if people have suggestions. We would love to be able to hire a bunch of people to just make these things, but we can't do that at the moment.
Okay. So I guess if I have time for one more question, so do you have connect points on the front and the back of the board, or just on the front, or the electrode?
Yeah. So right now, let me get to that. Right now, these are single-sided. This could be a double-sided board, but we have not tried double-sided bonding yet. So this is on one side. With the routing issues for 128 channels, actually, for a regular PC board, it is not clear that it makes sense to go double-sided because you'd have to route them in such a complicated way. And, I guess the other – oh, sorry, go ahead.
Oh, sorry. No, I was the question that I'm getting to is I'm curious if after you removed that shuttle, so on the electrodes themselves, can you record from the back side of the electrode on the side that you pulled the shuttle out?
Yeah. So yes. So in principle, when in fact Livermore has made double-sided electrodes, and we have tried them, it's not the issue for us is that that's also where the stiffener is. So we're probably doing more damage on that side of the electrode.
Yeah, I was just curious if the neurons would relax back into the electrode.
Yeah, we've gotten some signals from the back side of double-sided electrodes, but they weren't quite as good. But those were with big stiffeners in an older iteration. I'm sorry, I misunderstood. But so I think plausible. The issue for us here is that we're actually limited by the resolution currently of the photolithography because Livermore doesn't have an E-beam set up. So we're really trying to figure out, okay, could we make devices like we're showing here, but double-sided and much higher density? And so that's where we're hoping to go in the future.
Sure. Great.
Cool. So before we move on, I wanted to check in with Lynn to see if there were any questions from that side as well. You might be muted. We can't hear you, Lynn. Not sure if you're muted on your end. There we go.
Okay. So, great talk. So my first question is, can you elaborate a little bit on how you're going to do the simultaneous imaging with the unique recording?
Yeah, so again, a really great question. So one nice thing about these electrodes, and we've actually talked with some people about this, like Payman Golshani at UCLA, is because these electrodes are flexible, you could imagine, like in a two-photon setup, you could have a two-photon microscope, coming from the top. You could put the electrode in before you put in your window and then just bend it over. And so that actually has the possibility then of being able to have sort of electrophysiology and the high resolution measurements there on top of imaging at the same time, and that's where I think the flexibility comes in. It would be actually fairly trivial to combine this with the endoscope imaging. It's just a matter of getting those two data streams into the same computer and temporally synchronizing them. But those are all things we're sort of starting to think about.
Right, because I think if you have that ability, you can actually validate those phonic imaging data, right? I don't know how there's any drifting of this electrode in the brain. How can you validate, when you record from 18 neurons, for 100 days, how do you know which signal from which?
Yeah, no, that's a really good question. I mean, presumably if we could simultaneously image and do electrophysiology, we can cross-correlate them and ask, when do we see a signal from one and the other? But this is a broader question of, one of the wonderful things about electrophysiology is you put this small thing in the brain and you get neural signals and it does less damage. The problem is, how do you know who you're listening to? Actually, a colleague of mine and others have started thinking about the possibility of leaving these electrodes in place and doing electron microscopy afterwards. So slicing the brain with these electrodes in place, and then or some other high-resolution imaging to actually look at which electrodes or which neurons are physically near the contacts. And the question is, would that be good enough to be able to reconstruct which ones were giving the signal? And we don't know the answer to that, but I think it's another exciting possibility.
Okay, that's great. Yeah, that's all my questions.
Okay. All right. Well, thanks, everyone.
Thanks, everyone. Now we're going to go ahead and move on to the next talk. We're going to turn over the presentation to Ehud for his talk more on the optics side of things.
Okay, fine. So it's very good that we chose the order that we did. Loren gave a fantastic introduction to one of the big challenges of what we want to be able to do in studying the brain. And the work that I'm going to describe to you represents a complementary kind of technology, and it's complementary in two ways. One is that it uses imaging from the very beginning because of the advantages that you get in knowing which cells you're looking at and what processes you're looking at, although there are downsides in the sense of the speed of the signals being much, much slower than when you do electrical recordings of action potentials. The other is that much of the emphasis in the study of the brain so far has been on looking at action potential firing or manipulating that, and what I want to do is focus on synaptic transmission between neurons because that's the site where plasticity happens and where changes in the strength of how cells talk to one another mediates learning and memory, gates the different groupings of cells to operate at different times, which underlies switches from one kind of behavior or perception to another. And so I'm going to describe to you two sets of projects, both funded by the Brain Initiative, one on imaging, which involves two colleagues here at Berkeley, Hillel Adesnik, who works in mouse cortex, and Kristen Scott, who works in the fruit fly, and another one that's complementary to that on manipulation of synaptic transmission and plasticity, which is done collaboratively with another colleague here at Berkeley, Rich Kramer. And let me just, by way of introduction, show you one of the things that's so beautiful about the brain and that makes it so hard to study. So here's just a very sparse view obtained over 100 years ago by Ramon y Cajal of just one piece of the brain. And the reason that you can actually see the wiring diagram here is because he's using something called a Golgi stain, which just happens by magic to only stain every thousandth or so neuron. And so even though you can imagine if you saw every neuron, this would be a mess. It would just be a black mess because they're packed so much one on top of the other. But if you see only every thousandth, then you can see that even though these don't look exactly alike, they are of the same type. Their cell bodies are located in the same layer. They have dendritic ramifications of similar kind. They send axons in a similar kind of way. And by doing this and looking at different cell types, he was even able to come out with some suggestion about what connectivity looks like. And what we want to be able to do is understand the processes that mediate sensation, perception, integration of signals, and changes in the strength of communication between different parts of the brain that happen at contacts, the very fine contacts, let's say between this axon and the dendrite that you may not even see here of another cell type. And the question is, how do you do that? How do you visualize it, and how do you manipulate it in a way that allows you to test what it's actually doing? So first, before I tell you too much about that, let me just tell you about the fact that this is not simply an on-off binary kind of thing. There are many, many different chemical transmitter types that mediate signaling from one neuron to another. And here's just a list of many of them. The standard excitatory transmitter is the amino acid glutamate. The most common inhibitory transmitter is called GABA. But there are many, many other transmitters, some of them actually co-released by the same synapse. And that makes things very complex because they act in different kinds of ways for different purposes. And it's even more complex than that because there you are. Because if we just look at a cartoon of what an excitatory synapse that releases glutamate would look like, the glutamate is packed into these vesicles, about 1,000 molecules per vesicle. An action potential that arrives here produces the release of the transmitter from the vesicle into the extracellular space, where it actually can activate different kinds of receptors on the postsynaptic cell. There's a little projection of that cell called the dendritic spine. And it's complicated because you can see there are many, many different receptor types. 3 classes of ion channels, and 8 different classes of G protein coupled receptors called mGluRs. This is just what you see at the glutamate, the excitatory synapse. Each one of these different ion channel types and the different types of these work differently. They couple to different kinds of things, and they're each of them subdivided into a variety of subtypes which operate differently. So you can imagine that if you try to figure out what the synapse does, you would want to be able to know how signaling happens through this receptor versus this receptor. One reason that would be important is that activation of NMDA receptors is one of the most important ways of controlling the strength of the synapse for long-term, in something called long-term plasticity, because it regulates, among other things, how many AMPA receptors are on the surface. The more sensitive this postsynaptic cell is to the glutamate released from the presynaptic side. But there are many kinds of these, and there are many kinds of these, and these are not just located on the postsynaptic side, they're also located presynaptically. So the transmitter released here doesn't just act here, but also back here to control further release. And so if you had a drug that affected NMDA receptors, you could not point it at a specific subtype necessarily or specific location. And there are glutamate receptors also on these glial cells called astrocytes that also participate in controlling the release process because glutamate will activate them, and they can release, among other things, ATP and glutamate that can act back here or here. One short way of describing this is a mess, right? It's very hard to figure out what's going on with simple drugs that you wash and will cover the entire brain and that don't distinguish very well between the receptors, and you can't turn on and off very easily. That's the nature of the problem. Additionally, these structures are quite small, And so would you ever be able to visualize the effect of the release of this glutamate set onto some of these receptors? We want to do both things. Here's one of the problems. If you try to image neural activity, let's say you want to do what Loren was just describing at the imaging level, there are things called GCamp. GCamp is a calcium-sensitive version of green fluorescent protein, and you can load up cells with it, and when you want to just look at action potentials, you can look at the cell body, and the cell bodies are sufficiently distinguished from one another and fat so that you'd be able to image the firing activity here quite easily, even when GCamp is expressed so densely in this case, the brain of a zebrafish. But if you want to look at what's happening at the synaptic connections, you have to follow your way down these fibers into these extremely packed, intricate places where synaptic connections occur, and you can see you enter a tangle that you can't distinguish who's who anymore. So one question is, can we distinguish that? Is there some way of being able to do what Ramon y Cajal did, the magic of sparsity, in order to be able to look at just a few things at a time and distinguish them? And the version of it that we generated was a fluorescently activated, a light-activated version of GCamp. The protein is engineered so that when you shine light at it, it becomes fluorescent, and only the cells that you shine the light on can actually then be interrogated and followed. So here's an example. Many neurons packed in a culture. You can barely see them in a big, big tangle. But if you just give a pulse of light to one of those cells, it ends up getting filled, and the GCamp diffuses out. The fluorescent version of the GCamp now diffuses out all through the cell, and then that's the only cell that you need to record from. So here is an example of what you can see in that cell. You can see those cell-wide activity. Those are the action potentials. And here you can see that single action potentials can be detected as recognizable blips in just the way the normal GCamp is. You can do that with a slightly faster or slower version of these. And now what you can do is work your way through the circuit and say, I want to know what the connectivity is between several different cells. I photoactivate in cell 1. It fills. Now I know exactly how that cell looks. I pseudo-color it just to make it distinguishable from anything else. I now photoactivate cell 2. It fills. The places where they cross each other are sites of potential synapses. I can photoactivate another one after. And then I can correlate cell-wide activity here, let's say, which is due to action potentials, with local activity of calcium that's in, let's say, cell number 2, just at places where an axon from cell number 1 crosses a dendrite from cell number 2, a site of potential connectivity. And whenever there's good correlation, let's say between the action potentials that are in the red cell and local responses in the other cell, you get good cross-correlation coefficient. Here's a gradation of cross-correlation coefficient that actually tells you even what the synaptic weights are, how relatively strong the synapses are. So this is one solution, and we're now in the process of putting that with the work of Hillel and Kristen to look at the central nervous system of the mouse cortex and the fly brain. But the signal-to-noise there is not very good, and we'd really like to be able to go down to the detection of the single quantal events, the basic, most minor, simple, unitary synaptic transmission event that results from one synaptic vesicle dumping its contents into the synaptic space and activating postsynaptic receptors. For that, what we're doing is we're taking the GCamp and we just simply target it to the density of proteins located around the glutamate receptors here, taking advantage of the fact that those glutamate receptors actually let calcium into the postsynaptic cell. So what does that look like when you do it? Here is an example of a kind of recording that you see when you just give single action potentials, 2 axons innervating one muscle cell of a Drosophila larva. This is a glutamatergic synapse, very, very similar molecularly to our synapses in our central nervous system. And you saw those blips, and let's see what those blips look like in greater detail. Here is spontaneous blipping, which happens because of spontaneous fusion of vesicles to produce quantal transmitter release, just single synaptic events. You can see this at one site and at another site. Over time, these are groups of 50 milliseconds, so from 0 to 50 milliseconds, 100 milliseconds, you can see just a beautiful response locally, and this is what it looks like over time. The quality of the signal-to-noise here is as good as any electrical recording that you can imagine, but now it's spatially defined exactly at which synapse it occurred. So it allows you to do something that could never be done before, to figure out who's who when you're looking at a very complex network of inputs, let's say 1,000 inputs, or as many as 10,000 inputs to a single cell. This is what you would record electrically, a series of blips due to quantal synaptic events that happen spontaneously. But how do you know where these came from? Well, if you image at the same time, you get these local spots of activity, and you can now relate the one to the other, and you can basically say that electrical event was due to that synaptic event, so I know that this location produced that blip, that electrical blip. And you can basically unpack now over the entire synaptic input series where the synaptic transmission is occurring. The beauty of this is that it gives you a measure of one of the things that's been the most elusive when you try to measure it actually in a real circuit, and that is how strong is the synapse. You simply look at a spot when you're stimulating electrically, and you ask yourself, what fraction of the time do you have an event here? And what you see are zeros and ones, so now we are binary. And in this case, 40% of that presynaptic action potentials produce the postsynaptic response. So the probability of release is 0.4. Here it's double that. Here it's intermediate. And you can see these are different sites literally on the same axon. It gives you a view of a level of complexity of synaptic transmission that had not been previously appreciated, and a directness for measuring synaptic strengths that had not been previously possible on a synapse-by-synapse basis. The thing that's remarkable is that you can do this actually in vivo. So here is a maggot, and as you all know, who've seen maggots crawling around in places that make you think yuck, they're very opaque. Nevertheless, in the intact larva, which is somewhat constrained because it's packed into a very small chamber here, you can actually see the individual synapses as a very low magnification. So it's a very large part of the animal, and you can see waves of transmission traveling down, and during those waves, you can see what's happening at the synapse. This is a very low mag, so you can barely see what's going on there, but when you blow it up, you can actually detect single quantal events, even though it's in vivo. So what I told you is that we can image activity and synaptic transmission, and the key is either using photoactivation to sparsify, so we get a Cajal-like view, and we can do functional connectivity mapping using that, and we can look at greater detail with quantal resolution using these synaptically targeted GCamps, where we are able to measure directly not just the size of a response to a quantum, but the probability of release, which is one of the major things that's regulated during plasticity. Okay, so how would we be able to get something that is actually complementary to this, to not just measure synaptic transmission, but to actually control it? As I told you before, we don't simply want to control the release of the transmitter, because the release of the transmitter, the consequence of it is that it activates many, many different receptor types in all these different locations. So in a sense, if we want to really control the system, we want to do it at the level of the receptor, and ideally we'd want to be able to either activate that receptor or block that receptor in a very, very temporally and molecularly specific kind of way. So let me show you how we can do that. Imagine if the transmitter was not simply floating around, but was attached to the receptor itself via a string. And that string had a little connector which could be switched back and forth between two different configurations in response to light. In one case, it makes the transmitter stick out into the water. In another case, it turns a corner and sticks straight into the binding site, where when it binds, it activates the receptor, and if it's a receptor ion channel, it opens the channel and allows ions to flow into the cell. So here is what a photoswitch looks like. It has an attachment site that attaches here. It has the photoswitch, and it has the glutamate, in this case, hanging on the other end. And even a very short pulse of light, much shorter than an action potential, is enough to fully activate receptors, which, if left activated, desensitize in the normal kind of process, which is exactly what you'd see if you left glutamate on for a long period of time before switching them off, except if it were just diffusing glutamate, you couldn't control where it went, who it activated. In this case, you control it fully and in very, very short pulses of time. So the thing that's nice about this is the system actually works in complex settings. So imagine a case where you have an animal that's an animal model of a blinding disease called retinitis pigmentosa. This is a blinding disease because it kills the photoreceptor cells of the retina. The rest of the cells of the retina remain alive, and if we put our photoswitched channels into the output cells of the retina, the retinal ganglion cells, and use a version that a single wavelength of light turns on and it then spontaneously turns off in the dark, then you can do the following thing. If the receptor is there, but there is no photoswitch, you get the mouse is still blind. If there is a photoswitch, the mouse becomes sighted and can tell the difference between a flashing light and a steady light. So this is after training for several days. Mice swim. They swim very well. They don't like swimming. They'd rather be in dry place. There's a hidden platform here that will allow them to raise themselves up over the level of the water, and it's cued by the flashing light, but they never learn where it is because they're blind. However, if you've given them the photoswitch, then after the same amount of training, you can see that they've learned very clearly where it is. This can work also in the cortex. Here we have this put into the primary visual cortex, the input layer of that visual cortex called layer 4, and you can see in a single unit recording, so imagine Loren to a poor person, right, instead of 1,000 channels, just a single unit being recorded from, but on multiple trials, every time the light is turned on, activity turns on, it remains on in the dark because this is a bistable switch until you switch it off with an opposite wavelength of light. So now that's very nice that works. We would love to be able to do this on the receptors that control synaptic plasticity. And here's a case where this is done for NMDA receptors, key controllers of synaptic strength, as I mentioned before. This is a version of photoantagonism, where when the photoswitch turns in, it blocks access to the binding site but doesn't actually activate the receptor. And you see that you get, when you activate it with a drug called NMDA, which is what gives it the name NMDA receptors, then the photoswitching interrupts the response. The response would look like this, but little pulses of light now undo that and give you a partial block of the response. And you can also do photoagonism, meaning photoactivation, and now the pulse of light actually turns the current on. So let me just show you two examples of how that works. This is an example from the hippocampus, which is one of the main seats of producing long-term memory and memory consolidation to go from minutes to very, very long-term of hours, days, weeks, and years. When you record from one neuron in the hippocampus and you stimulate the axons that innervate that neuron, then you can, if you give a buzz of high-frequency stimulation, produce long-term potentiation, LTP. And can we photoantagonize now, meaning photoblock, and prevent that from happening? And the answer is yes. When this is done at the wavelength that doesn't activate, so the thing is pointing out in the breeze, then the buzzes produce a strengthening. Here is the size of the synaptic response, and it doubles in strength. But when you now photoswitch, so this docks in there, does not activate, but prevents the normal synaptic glutamate from binding, then you can see you block the induction of the long-term potentiation. But you can do the opposite too by photoagonizing, and this is just one view of that. You can do it even with the precision of a single synapse because you can point light at a single dendritic spine where the receptors are, the postsynaptic receptors. And when you do that, the spine actually swells in size. That's part of the way that spines get stronger, and you can see that swelling. It goes up to a high level and then settles at a higher level than original, and it's confined only to the spines that were actually illuminated. So you can produce long-term potentiation very, very specifically and locally. You can block its induction as well based on whether you photoagonize or photoantagonize. And this works not just on the ion channel receptors, but also on the G protein coupled receptors. And there's an entire family of these, and you can see you can engineer every one of them because it really is modular engineering. Okay, so let me just summarize what I showed you there. This is called chemical optogenetics. It's work that Rich Kramer and I have worked on for some time, and we've now taken it to a point of development where we're putting it in vivo and using it in vivo in mouse models and in fish models. And that's what's being supported by the grant from the Brain Initiative. I spoke to you about the glutamate-activated ion channel receptors, the NMDA receptor in particular, and the metabotropic G protein-coupled receptors responding to glutamate. Rich has been focusing on the inhibitory synapses that are working with GABA and on the modulatory synapses that are gated by acetylcholine. But this technology also works with ATP-gated receptors, with proton-gated receptors, and with ion channels that work at synapses that are gated by voltage, calcium, and many other things. So I think that the nice thing about what Loren presented and this is that it allows us now to be able to combine things that allow recording from many, many cells to know when cells are active and understanding of synaptic connections between the cells and the ability to measure the strength of the synaptic connections as well as manipulate them in order to really understand how neural circuits operate and are reorganized by experience in order to gate behavior. So I can stop there and take some questions, and I'll just point out that one of the moderators, Yantian, has an alternative strategy that she uses to detect, to image synaptic transmission, where she actually directly images the synaptic transmitter, which is a very nice complementary method. Okay.
Cool. Thanks, Udi. And that's a good segue. We'll go ahead and turn it over to Lin Tian to start the discussion period.
Hey, Udi, this is a great talk. So my first question is about the photoactivable GCAM. Can you, no, it's a great tool to have, but can you give me, just comment on the capability to be able to use that in vivo for functional, connectivity mapping? I understand, because you have to highlight each cell, it's kind of, because on one hand, we want to really record as many neurons as possible, right? But if in order to understand the functional connectivity, in vitro, which is you only have a few cells to manipulate. But if you want to do this in vivo, I just kind of want to know what's the utility and what is the best example. We really need this, use those two to do the functional connectivity, rather than doing you have heterodopsin and you can simulate on one region and then record from the other region.
Okay, so I'm sorry. So you're asking about the optical manipulation of synaptic transmission compared to channel rhodopsin, not about the imaging of synaptic transmission. Is that right?
Yes, that's it.
Okay, so when we do the optical manipulation, many of the same things that hold for channel rhodopsin hold here, which is you're constrained by the ability to point the light and the distribution of the antennae that are sensitive to light. So as with channel rhodopsin, you can target the expression of the receptor just to certain neurons genetically, and you can target the light by pointing it. Light can be sculpted in 3 dimensions in a variety of ways, and the question is, how do you put in the difference between channel rhodopsin and this, in part, is channel rhodopsin is completely a different kind of beast It's orthogonal biologically. It's an added-on thing that comes from a microbe. Here, we're actually manipulating the native receptor. That's both an attraction, and it means that if we just take a virus in order to express it, and we express it on top of the native thing, then we will be overexpressing the receptor. In some cases, that's not a problem because, as with NMDA receptors are made of two different classes of subunits. So let me just show you. There is the GluN2A, or GluN2 subunit, which binds glutamate, and the GluN1 subunit, which binds glycine or serine. These come in a one-to-one configuration. There are actually two of these and two of these in each receptor. And GluN1 is an obligatory component, and it's rate limiting to how many receptors you can have. So if you engineer one of these and express it in a cell, and you overexpress it, you won't get more receptors than you would get normally because they will simply outcompete the native receptors to join the GluN1 subunit. So that's kind of a poor man's, but very convenient, knock-in. However, of course, in some cases that doesn't hold, and even in this case, there's an advantage to knocking things in, and knocking things in ways that can also be sub-targeted to specific locations. So, in fact, one of the things that we're doing now is we're going from these overexpressed conditions to create these knock-ins. The knock-ins are actually critical for this kind of work, whether it's working with the NMDA receptors, the kainate receptors, meaning the ion channel receptors, or the metabotropic receptors, the GABA-A, the GABA-B, and so on. So knock-ins allow you to do that in a selective way. We hope, of course, that knock-in technology is going to become radically easier with CRISPR. It is much, much easier in some organisms. It's working fantastically in flies. In mice, it works quite well. In zebrafish, it's still a struggle to do knock-ins if you actually make significant changes. But for single amino acids, hopefully it's not going to be too hard there, too.
Okay. So do you imagine that you can take those kinds of manipulations to the human application to be able to treat blind in the future?
So to integrate, you mean the opsins with our chemical optogenetics?
Yeah, so to be able to apply to be able to, have the application in the human.
Yeah, so integration actually across a variety of things is something that's very, very attractive. Well, one thing you'd like to be able to do is you'd like to be able to integrate the ability to, let's say, photocontrol a specific type of NMDA receptor with optical detection of synaptic transmission, or optical detection of neuronal firing using cell body-targeted GCAMs. In some cases, you'd like to be able to control an NMDA receptor and a GABAergic receptor, so you need to have different kinds of ways of attachment so they don't cross-react, the photoswitch here for the photoswitch there, and you want to be able to have different wavelengths so that they can be controlled separately from one another. So what we're doing is we're trying to figure out ways of packing into color space, spectral space, things that can read out and read in, and chemical strategies of attachment that also can be complementary. And so far we've succeeded in doing two different photoswitches, separated by different kinds of methods of attachment and by different spectra with electrophysiological recording. And we're working with that now to be able to also do optical recording at the same time. It's a challenge. It's even a challenge still to get a channel rhodopsin that will work with a GCAMP because they tend to cross-contaminate. So I think that that's a general challenge for the field. There are good strategies for how to solve them. I think we're on the way to doing that.
All right, great. I have one more question about the photoactivable GCAM. So how that compare, in terms of brightness, how that compare to the regular GCAM after you turn it on?
Yeah, so I mean that's a very good question. nothing that you've monkeyed with is ever as good as its parent. So I showed you that we're able to detect single action potentials, but the signal to noise is about 20% as good. So we do take a hit. You can see single action potentials here, right? So you can make the measurements, but the signal of the parental GCAM6 S or F is 5 times stronger. What I should say is that part of the effort now is to not simply deploy these with Hillel and Kristen in the fly brain and in the cortex, but to also make variants on these. That these are all green, even though the traces here are shown in 2 different colors. They're all green variants, but to make them into various kinds of red variants that are better. The red variants of any of these calcium sensors already are not as good as the green for the parental, and so it's that much more of a challenge. But actually, we're on the way. We already have something that works quite well in the red.
Yeah, it's still very useful too. Yeah, that's all my questions. Do you have any questions?
So I just have a quick naive electrophysiologist question. So assuming everything works out really well in vivo, can you use this technique across sort of the timescales of a behavioral mouse experiment?
So the answer, I mean, in both for the imaging and for the manipulation, the answer is yes. I mean, so for the longest we've gone chronically in mouse has been with the example I showed you for the retina, for the photo control.
Yeah.
And that's something that we do over weeks and months.
Okay, great.
The thing I have to say is that the photo switch, that's for this application. The photo switch ends up attaching covalently to the receptor, which is located here. So when the receptor gets turned over, new receptor comes to the surface. If it has not recycled the receptor, it means that now it's not light sensitive anymore. And so with a conventional photo switch, we have to reintroduce the photo switch on once every week or so. We now have a new chemical strategy that allows us to do it once every 3 weeks because the photo switch actually just persists in the retina for longer, and it doesn't get destroyed by hydrolysis. We're making slow-release versions that hopefully will be working for months. So that's a limitation there, but otherwise it's extremely robust, and the cells tolerate it very well. That's for the photo control. In fish, the longest we've gone is 2 days, but these are larval fish. After a couple of days that we're studying them, they go on to develop and they become opaque, so we can't actually use light on them so easily anymore. With the photo imaging, right, of synaptic transmission, again, we've done it over a period now of days in the fruit fly, but it's days of larval development. The real question is what will happen in cortex, and that's what we're in the process now of trying to assess. So I can't quite answer that question yet. I hope that in a year we'll have good answer to that.
Great, thank you.
Cool. We're already quite a few minutes over, so I think we'll go ahead and wrap up this first of the webinar series. I'd like to again thank Udi, Loren for presenting, as well as Cindy and Lynn for serving as moderators. We're currently working on scheduling the next one in the series in the coming weeks, and we hope everyone will join for the next in the series as well. Thank you again, and have a good weekend.
Bye-bye.